Okay, let's talk about CRISPR. It is, without a doubt, one of the most incredible technologies out there. It lets scientists edit the very code of life. You've definitely heard the name, and for a really good reason. It's already performing what, honestly, looks like miracles.
And look, saying it's a game changer isn't an exaggeration. We're talking about a tool that went from a fascinating scientific curiosity to a life-saving treatment that was actually used to rewrite the DNA inside a living infant. I mean, that's the kind of power we're dealing with here. So what if I told you that even as CRISPR is out there making these miracles happen, its potential successor is already being discovered? And the craziest part? It's being pioneered by one of the key scientists who helped unleash CRISPR's power in the first place.
This is Feng Zhang. He was absolutely instrumental in figuring out how to adapt the CRISPR system for human cells, which is what unlocked its massive medical potential. But instead of just polishing the tool he helped create, he went back to the source, back to nature's code, hunting for something new. And you know what? He found it.
Now, to really understand this new discovery, you have to understand how Zhang sees the world, because his journey didn't start in a biology lab. It started with a computer. So growing up with parents who are computer scientists, Zhang was always encouraged to take things apart just to see how they ticked. For him, it was old PCs. But then, a 7th grade molecular biology class completely changed his perspective. He had this light bulb moment. DNA, with its own code and instructions, was just like a computer program. From that point on, his mission was set. He was going to learn how to tinker with the code of life itself.
This quote from Zhang himself just perfectly sums it all up. He doesn't see life as just a bunch of chemicals. He sees it as an information system. And if you can understand the code, well, then you can change the output. It's a really simple but profound idea, and it's the key to everything that comes next.
So armed with this code breaker mindset, Zhang turned his attention to this really fascinating defense system that you find in bacteria. He didn't invent it, he just learned how to harness it.
So what even is CRISPR? I know, the name is a total mouthful, but the concept behind it is beautiful. It's basically a bacterial immune system. Bacteria use it to find the DNA of viruses that are trying to invade, remember what it looks like, and then chop it to pieces if it ever shows up again. It is literally nature's own cut-and-paste software. And here's the basic playbook, in 3 simple steps. First, a little piece of guide RNA. Think of it like a biological GPS scans the entire genome for one specific target. As soon as it finds a perfect match, it binds on tight. And finally, an enzyme, which is basically a tiny biological machine, comes in and acts like a pair of molecular scissors, making a super precise cut.
And this is where Zhang's contribution was so crucial. See, while others discovered the system in bacteria, he was the first person to successfully engineer it to work inside the much more complex cells of mice and humans. That was the breakthrough that took CRISPR from being a cool scientific fact to a world-changing medical tool.
And the goal here, well, it's pretty much the holy grail of medicine. If we know that a disease is caused by a specific genetic typo, a single mutation in the DNA, we can use this tool to go in, snip out the mistake, and let the cell's own natural repair systems write it back to the healthy version. You're not just treating symptoms anymore, you are fixing the problem at its absolute source.
But even a tool as revolutionary as CRISPR has its limits. And for a mind like Zhang's, limitations aren't roadblocks. They're basically invitations to go find something even better. And that's exactly what pushed him to search for the next generation of gene editing tools.
So, where does CRISPR struggle?
Well, it's fantastic for diseases caused by a single genetic error, but a lot of conditions are way more complex than that. Also, just getting the CRISPR machinery to the right place, like deep inside the brain, is incredibly hard. And sometimes, its cut is more like using big scissors, when what you really need is a surgeon's scalpel. It can affect a few genetic letters when you only want to change one. So this sent Zhang and his team back to the drawing board, hunting through nature's code for another tool. And they found one. They called it Tiger. And get this, instead of finding it in bacteria, they found this system mostly in viruses. The very things bacteria use CRISPR to fight. It's a system born from millions of years of evolutionary warfare.
So how does this new challenger stack up? Well, while CRISPR comes from bacteria, Tiager comes from viruses. One huge difference is that Tiager reads both strands of the DNA double helix, which could make it way more accurate. It's like it's double-checking its work. It's also much more compact, which is a massive deal when you're trying to actually deliver it into cells. And it just might have that single-letter precision CRISPR sometimes lacks.
And just to give you a sense of the incredible science going on here, to even study this tiny TGR system, the lab has to concentrate it.
They do that with centrifuges that spin with a force up to 100,000 times the force of gravity. That is just a staggering number, and it shows you how high-tech this work really is.
Okay, so this is all fascinating science, but what does it actually mean for us? What's the real-world promise of a more compact, more precise gene editing tool?
Here's why this matters so much. CRISPR has been really effective in places like the liver, which is a pretty easy organ to target and where cells divide a lot. But TAGR's compact size could be a total game changer. A smaller tool is way easier to package up and deliver, which might finally let us get these therapies to places that have been pretty much out of reach, like the brain. And if you can get to the brain, you can start seriously thinking about treating some of the most devastating neurological diseases we face. Conditions like Alzheimer's, Parkinson's, ALS, and Huntington's disease. These are all illnesses where we know there's a genetic link but have struggled to find effective treatments. TAGR could be a whole new avenue of hope.
And this brings us right back to the man behind the science. For Feng Zhang, it's not just about the potential cures. It's about the pure, unfiltered joy of discovery, of asking a question that nobody in the world knows the answer to, and then being the very first person to find it. That's what it really means to push the frontier of human knowledge.
And so that leaves us with a pretty profound question. As scientists like Feng Zhang give us more and more powerful tools to edit the code of life, we are, in effect, becoming the programmers of our own biology. The question isn't really can we rewrite the code anymore, it's how should we? What rules are we going to write for ourselves?